Hello and welcome to the training video for the Mill Creek Alliance Water Quality Monitoring Program's Nitrate-Nitrogen Testing Station. Nitrate-nitrogen is a nutrient that is natural to aquatic ecosystems. However, surplus amounts of nitrate-nitrogen can enter the water from human and animal waste, decomposing organic matter, and fertilizer runoff. This can lead to increased plant growth and eutrophication. High levels of nitrate-nitrogen can also interfere with growth development in fish. The nitrate-nitrogen testing station involves the use of chemicals. You should wear the gloves and safety goggles provided for your protection. Should an emergency arise involving the use of these chemicals, please refer to the Right to Know station in the laboratory. On sampling Saturdays, field volunteers will collect samples from our sites and deliver them to the lab for analysis. These samples should be available for you to use when you arrive on site. When you arrive at the nitrate-nitrogen station, the following materials should be available for use. If anything is missing, notify laboratory staff. Equipment: Hach DR3900 spectrophotometer, nitrate standard solutions in 0.5, 1, 2, 5, and 10 milligrams per liter, 1000 microliter pipette, 200 microliter pipette, 500 milliliter wash bottle, white plastic test tube racks, waste container, a fine tip Sharpie and pencil, safety goggles, and the nitrate-nitrogen SOP. In addition, you should have the following supplies: Hach Nitrates TNT Plus 835 cuvette tubes, Nitrate TNT Plus reagent, 50 to 1000 microliter pipette tips, 2 to 200 microliter pipette tips, DI water, Kim wipes, nitrile gloves, and the nitrate-nitrogen lab data sheet. Review the SOP provided before you begin and refer to it while working as needed. If you have any questions, contact laboratory staff. Begin by writing your name and the testing date at the top of the lab data sheet. With the testing chamber lid closed, power on the spectrophotometer. The meter will complete a self-check, then go to the main menu. Press the Barcode Programs button on the main menu. This test is fully automated. From this point forward, once you insert the test tube into the meter will do everything else for you. You do not need to zero the meter. Label test tubes for all the samples you are going to run with the marker provided. Note that some site numbers on your datasheet may be missing if a site has been retired or changed, so mark tubes carefully. You will be running samples for a before testing standard series, each of our sampling sites, 5 field duplicates, 5 laboratory duplicates, and an after testing standard series. As this test is time sensitive, you should prepare samples in small batches that you can comfortably manage. 10 samples per batch is recommended. Your first batch should always include the before testing standard series, and your last batch should always include the after testing standard series. Stream water samples used for this test come from the larger 250 milliliter sample bottle. Bottles are kept organized in the main lab area. All lab volunteers share usage of these bottles, so you can pick up a small batch of bottles to work with, then return them and get another set. You will not necessarily be processing the samples in chronological order. For each sample, complete the following testing routine. Shake the sample vigorously to make the sample homogeneous. Place a new, clean, 50 to 1000 microliter pipette tip on the 1000 microliter pipette. 1,000 microliters equals 1 milliliter. Using proper pipetting technique, withdraw 1 milliliter of sample from the sample bottle and waste it into the waste container. This is done as a quality assurance measure. Then, withdraw another 1 milliliter sample from the sample bottle and dispense it into your appropriately labeled test tube. Next, place a new, clean 2 to 200 microliter pipette tip on the 200 microliter pipette. 200 microliters equals 0.2 milliliters. Using proper pipetting technique, withdraw 0.2 milliliters of nitrate TNT plus reagent and dispense it into your test tube. Cap the test tube securely so it does not leak, shake to mix, and replace it into the test tube rack. Take care when handling the test tubes at this point, as they will become hot to the touch. Start a 15-minute timer. You may use the timer on the Hawk machine or your own personal timer. Once the 15-minute reaction time is complete, you will continue to process each sample as follows. Use a Kim wipe to remove fingerprints or other impurities from the outside of the test tube, then insert the test tube into the meter. The meter will generate a reading. Enter this reading in the corresponding space on your lab data sheet. Remove the test tube from the meter, then reinsert it to generate a second reading. Enter this reading in the corresponding space on your lab data sheet. Move the completed test tube/sample to a predetermined finished area in your test tube rack. Continue this process with all the samples in your batch. Process the next batch using the same testing routine until all samples are completed. Please note, beyond the 15-minute reaction time, sample color may overdevelop and lead to inaccurately high readings. For this reason, samples must be read as soon as possible after the 15-minute reaction time is complete. Run small batches of samples for this reason. Do not attempt to run more than 15 samples per batch. Once all testing is completed, clean up the station as directed by the SOP.